Hello, my name's Erinn Fagan-Jeffries. I'm a postdoc at the University of Adelaide on an ABRS postdoctoral fellowship. I'm about 18 months out of my PhD, so I'm an ECR, and this presentation kind of comes from that perspective, so that's kind of the caveat. This was the question that Kevin threw at me: how will we discover and document the remaining hyperdiverse? So what I thought I would do was go through what are the hyperdiverse insect orders, at least in terms of species numbers for Australia. How many species are we actually talking about when we say that these things are hyperdiverse? What are the common issues, or what, at least from my perspective, I think are issues that I face that probably researchers working on the other groups would face as well? And then I thought I would look at my study taxa, the microgastroenterine wasps, kind of as a case study and go through my process and the current situation of how we go about describing new species, because I imagine that at least some aspects of it are probably very similar to how other researchers in these groups go about the taxonomic process. I thought I would look at what slows us down, again, because I imagine this is probably quite similar across the board, and then just throw out a whole bunch of ideas of what we could do to perhaps make this. These are the currently described species in the Australian Fauna Directory for the largest 5 orders of insects. So I'll let you read them at your leisure, but we've got Coleoptera at the top, Hymenoptera, then Lepidoptera, Diptera, and Hemiptera. So there are altogether about 70,000 insects described, with these 5 orders being the largest. Are not the true numbers of species. So these are the estimates of the true number of species, which come from the Chapman 2009 ABRS publication, and they got the information from the Yates et al. Paper in 2003. So a lot of these numbers are possibly underestimated, but they're kind of the closest thing we've got so far in terms of putting real numbers to these things. So for the Coleoptera, the estimates that Yates et al. Put out were 80 to 100,000 species. For Hymenoptera, they suggested there could be 44,000. I don't know how many people are aware of the paper that came out recently that looked at a lot of trends and suggested that really for every Coleoptera species, every beetle species, there's probably 2.5 to 3.2 times the number of parasitic Hymenoptera. So really, if there are 80 to 100,000 Coleoptera in Australia, we could possibly be looking at 250,000 Hymenoptera. It might sound crazy, but based on other studies, it's possibly, a real thing. With the Lepidoptera, their estimates are about 20,000, Diptera about 30,000, and Hemiptera about 11,000. So as you can see, there are groups that are hugely currently under-described. The common issues that I think taxonomists working on these groups possibly face are that lots of them are really small in size. So from the Hymenopteran perspective, lots of these parasitic wasps where you get all this diversity are really, really small. So this is a picture here of the family Mammaridae, which can be normally around 0.5 to 1 millimeter long. It also includes the smallest insect in the world. They're known as fairy wasps, and that's 0.13 millimeters long, a species from North America, I think. So these things are tiny, which obviously has a whole bunch of issues around it. Part of that is that as things get smaller, they often tend to be more morphologically conserved. So the characters that we would use to help look at relationships and to look at species boundaries become less and less and less. As things get smaller, for example, they lose wing veins, so suddenly you don't have those characters available to use in your species delimitations. There's lots of studies looking at pseudocryptic species and true morphologically cryptic species. Another issue that I think happens in this group is that there is previous taxonomy. but it's not always correct, and this can actually sometimes cause more issues than if there was no taxonomy and we could kind of just start again. A lot of that previous historical taxonomy has been done by overseas researchers, so the types, for example, are in overseas institutions. They're often not in good condition. We don't have molecular information for them, which makes things really difficult. We're often talking about a lack of specimens. So I know when I am working on a group and trying to describe species, I'm often, working with maybe 3 to 10 specimens per clade or per potential species. So, and we also often are looking at lots and lots of singletons, which can make doing the taxonomy tricky. And these groups only require really specialist expertise and specialist facilities, which partly has to do with their size, right? So really tiny things are difficult to do taxonomy on by, for example, provincial institutions that don't, say, have imaging, sequencing, high quality microscopes, the things that you need to be able to do the descriptions on these groups. Another issue that I think is probably well known is that the workforce is not proportional to the size of the taxon groups. So we have a lot more people working on, say, vertebrates or flowering plants, or even in insects in Lepidoptera, despite that being one of the less diverse of these orders. We also generally have no ecological biological information for a lot of these specimens that we're using to do taxonomy on, which means we don't have that kind of extra information to help support species limitation or any of that kind of thing. Right, so the microgastrines, for anyone who hasn't heard me talk about them before and isn't familiar with them, I think they make kind of a cool case study for how we go about describing these really crazy diverse groups. They're quite small, they're 2 to 3 millimeters long, they're endoparasitoids of Lepidopteran larvae. There's nearly 3,000 species formally described globally, but estimates suggest that we're probably looking at up to 40,000. In Australia, there's about 150 species described, and using some ratios with the number of Lepidopteran species and how many parasitoids in other parts of the world have been reared from different Lepidopterans, we're looking at possibly 1,500 species at least of microgastrines in Australia. So we've currently described the history of taxonomy in this group is a fairly long one. Catesia glomerata, which is an introduced species, was described by Linnaeus back in 1758. The first native species was described in 1900 by Ashmead, and then there were 2 workers who kind of did the bulk of the descriptions for the Australian fauna before the modern work. So Wilkinson in the 20s and 30s and Nixon in the 60s. And then Andy Austin and his lab, myself included, have been working on this group since the late 80s, kind of in fits and starts. This is kind of the history of where the taxonomy is at the moment. If we take myself as an example of an ECR who has been doing taxonomic research since I started my PhD in 2015, these are the numbers of described species that I've been able to publish since then. If we plot that on a graph, using Kevin's idea that he threw out of naming all species in a generation, and it's arbitrary, but he mentioned 25 years at one point, so I use that as my projection in 25 years. How many species might I describe if I keep going business as normal compared to the number of species in Australia that we think might be out there? So I'd like to think I get more productive as I get further into my career. But I'm obviously even a little bit more productive than I am now. I'm not going to get anywhere near that 1,200 species, and that's just one subfamily of parasites. I'm not going to be able to do this if we keep going as we're going. So I thought what I would do is take you through my normal process for describing species, because hopefully that will help identify where the bottlenecks are. So it obviously starts with specimen collection in the field or going through museum samples. I then always start with DNA barcoding. So as I said, the morphology in this group is often really conserved. It's very difficult to work with with specimens starting with a coarse morphosort. So I start with DNA barcoding. And at this point, I lose specimens and potentially species from the pipeline because I don't tend to treat specimens that are unable to be barcoded. I only work with specimens that I am able to get molecular information for. Now, we're quite lucky in this group that we have a divergence threshold in CO1, which has been quite well substantiated in some really big rearing studies that have gone on in Costa Rica. So we have this 2% threshold, which seems to work quite well. Then, of course, as everyone would, we go through that specimen sorting, pinning, labeling, all that kind of work, which is obviously quite time consuming. At least in the university system, we don't have technicians or anyone to assist with this. So all this is done by the taxonomist. At this stage, any species or clades that have come out of the molecular species limitation that are only single specimens or that only have male specimens, because in this group, the females are very important for characters, and it's generally, I guess, looked down on in taxonomic publications to publish things based on single specimens, for perhaps obvious reasons. These clades are kind of lost, or these species are lost from the pipeline. The morphology is then examined at a fairly coarse level. I then go back to the type specimens and examine them wherever possible, and try and match them up to any of the clades that have come out of the molecular delimitation. Are any of the described species present in the material that I'm looking at? And this can be a really difficult thing because obviously we don't have molecular information for these type specimens. So where we can rule out that clades based on morphology don't match up to these described species, we then find, we make the species decisions and we find morphological characters to delimit molecular species. So if we can't separate them from described species, then I generally take the conservative option of not wanting to create synonymies, and I don't treat these. Morphological descriptions then occur. We measure lots of different things under the microscope on multiple specimens that are not necessarily used in the diagnoses, and that probably are not particularly useful characters because there's so much variability in how researchers measure them. So how I measure the length of the first metasomal tergite is probably different to how someone else is going to measure it. So you could say that perhaps these things are a little bit arbitrary. We then are normally encouraged by reviewers to write a morphological key, which can be very difficult. And then, of course, it goes through the whole publishing pipeline, which everyone knows how crazy and time-consuming that can be. So these are the locations where I definitely lose specimens. And obviously, also at the start, when we're collecting specimens, if we're talking about describing all species in a generation, we can only describe those species which have specimens collected. So how broad and expansive that actual field component or museum mining component is will obviously play into how many species. When I presented this at the very reduced meeting that happened in Adelaide last week, I was asked which bits do I think are the most time consuming, which I found are a super hard question to answer because everything takes a really long time, other than the molecular work, which is really fast now. But I think writing the keys, when I've done that, that can just be so frustrating and time consuming. Measuring, lots and lots of characters on multiple specimens can be super, super time consuming. But also this is probably what I think often takes me the longest time, is where I have got quite a bit of historical taxonomy and I need to really rule out that the described specimens where I don't have molecular information are not present in my material can take a really, really long time. Right, so what I thought I would do is go through each of these parts and look at possibly some ideas for how we can make these more efficient, at least from my perspective in describing microgastrines, but I imagine they're probably much more broadly applicable. So to make this more efficient in terms of specimen collecting, we can have better sharing of specimens between individuals, better coordination of bulk sampling, especially on programs like Bush Blitz. This still isn't happening. This really broad bulk sampling on, centrally funded expeditions, which I think is crazy. And some of this is already happening, especially the better sharing of specimens. So I know there's now a much bigger push for us to be sharing material, and I think this is only going to get more and more collaborative. But this bulk sampling stuff by institutions can maybe have a lot more investment and community investment into it. But of course, we can't expect institutions to be bulk sampling and looking after this material without some kind of funding investment, because I know how pressed for space museums are and how difficult it can be for them to store things properly. So, for example, if we are asking institutions to do bulk malaise trapping and bulk pitfalling and all that kind of thing on, for example, bush blitzes, how can we allow them the facilities to be sorting these to order, to be storing them in cold, clean ethanol, and to be maintaining these collections for future use? I then do the DNA barcoding, and already new technologies are making this much faster, but it's still not cost effective to DNA barcode lots of old specimens. So, there's so much pinned in collections which I don't access because I don't have the funding to go through the process of getting the molecular information for these pinned specimens. But I do think that in the very near future, this will be something that's much easier to do and much more. I then go through molecular species limitation, and I guess the perhaps crazy idea, but and it's an idea that needs to be talked about, is do we ever have the pipeline stop here? Do we delimit molecular species and publish their names and stop here in terms of getting names on things, getting that information out to the public with the knowledge that the morphology can go back and happen later? But of course, if we don't look at morphology and we don't have molecular information for every single type, how would this work with existing taxonomy? Then the specimen sorting, pinning, labeling, all that kind of stuff happens. Can we try and get some more automation happening, some better sharing of techniques and facilities to do this? More acceptance of having QR codes on a pin instead of having a label that you've typed out and printed and cut out and that sort of thing. Like, how can we get this happening in a much more economical and faster way? Can we have more funding for technician positions in universities and in museums to kind of get this process being a better use of the taxonomist's time? Can we have more acceptance of describing species based on single specimens? Do we want this? Because if we're talking about describing every species, then we need to plug these holes in the I then go through, look at the morphology at course level. Unless we are stopping that pipeline at the molecular species, it's kind of hard to make this faster without disregarding the morphology. I'm not suggesting we should do that. I think there would be something a little bit sad about taxonomy if we didn't look at organisms. That's how people relate to the environment. That's why taxonomists get into taxonomy, is because they love looking at their taxa. So I'm not suggesting we want purely molecular species, but it's something that needs to be thought about in some groups. And also, we need to think realistically about how people who are the end users of our taxonomy are going to identify species in 10, 20, 30 years. Are they really going to be examining specimens, or are they much more likely to be sequencing them? Which information is more important for diagnostics? We then do this process of examining type specimens and making the morphological species decisions. Something that would make my process a whole lot faster, and I imagine a lot of others as well, would kind of be this whole community investment into sequencing type specimens, both those in Australian institutions and those overseas. So I know ANIC is embarking on this really amazing project to do genome skimming of type specimens, but I'm kind of curious about how that is going to be prioritized, and perhaps the best way to see really quick return of that is prioritizing taxon groups that have current expertise and projects. And we then do the morphological descriptions. Can we reduce these descriptions? Can we have more acceptance in publications that instead of having a description that is based purely on what's been done for 100 years, instead we just have a really clear morphological diagnosis, images and sequence info. And do we want this diagnosis to be morphological? Do we want it to be molecular and morphological, or only molecular? Which goes back to the question I've been throwing out the whole way. We then write a key. Are morphological keys going to be useful in the future? Will the people who are writing these keys for really use them? Are they going to be examining specimens? Are these keys even useful for non-specialists, or are we really writing them for the next person who's going to be working on this group? And, there's such a time sink, at least in my experience on writing these keys, because of the fact that I can't use kind of auto-generated keys that come from measuring characters and plugging this all into Delta or Lucid or something, because those characters are not particularly useful for species limitation. They're too variable, they're too vague, they're too different based on who's measuring them, too kind of subjective. So this process of finding characters that work for keys is really time consuming and maybe really not particularly useful. Get the paper writing and formatting and publishing a bit faster, I think, will be one of the easiest things we can do, but we need to make sure that we are really maintaining the rigor and peer review and trust in the process. And one of the easiest things that might be able to happen is have a journal, for example, with more relaxed formatting submission requirements. Instead of trying to type out all your examined material in exactly the right format that the journal wants, we just have, a link to an online database where all this information is already uploaded.
So thanks, Erinn. That was a really good presentation. I really liked that. I really liked the way you went through with that very methodical sort of flow chart about how you do things. And since seeing your presentation, I've sort of thinking in the same way myself and using your flow chart as the basis for some others. So that's really good. And it's not often that the process of taxonomy is so neatly set down. So that was great. So I just had a few questions that came to my mind as I was watching your video, and I'm very much hoping that other people who watch it can ask you questions through the comment box at that's on the page under the video. So my questions were, so I'm intrigued as a botanist that you are working with a taxonomic group where you so often describe species on the basis of very few specimens. Mm-hmm. I've sort of got two questions about that. The first one is, how much of an issue do you think that is? How much of a handle can you get on the likely morphological and genetic variation in a species? And what are the consequences of that for taxonomy?
I mean, I guess it's a slight issue in that you never really know the extent of the differences and the changes in the morphology within the species. But I think it comes back to that idea that the species is a hypothesis. So as more specimens get collected, then the definition of the species is possibly going to change from that original description, which is based on, say, 2 or 3 specimens. But if we wait until we have more specimens to describe these things, then they're never going to get described. So it's kind of a case of, we can look at the issues, or we can look at, well, that's just how it is. So we deal with it and work with what we've got.
Yep. Hey, I should have explained to people watching this that you're at home because of the coronavirus, but you've got a very nice dissecting microscope in the background of your more imported Malays trap samples in your living room, or
Yes. Yeah, borrowed from the university, so keeping working.
yeah. Well, here is evidence to Andy Austin that you're working. Andy, I can tell you that Erinn turned from the microscope when we started this Zoom meeting. Great. So the most extreme case of
Mm-hmm.
having very limited specimens is, as you said in your flow chart, if you have only a single specimen, it drops out. You won't name something.
Yeah, I guess that's something I've done so far is that I've hesitated to name things based on one specimen. And to be honest, that has to do more with what I thought the tradition and the culture was around that in the field, that was something that you weren't supposed to be doing, I guess, for all the reasons we talked about with the issues of describing based on just a few specimens, is that how do you look at the patterns and the variation within a species if you've got one specimen. But I think this whole process of looking at the Taxonomy Australia mission, to me, has really shifted my view on that. And if I've got a specimen that is distinct morphologically and that I have DNA data for, I feel like if I felt like it would get through review, I would be much more willing to describe those sorts of things.
So the issue of getting through review is the big one at the moment. You know, there are sort of communities of practice, and some things are frowned upon in review. That
Yeah, and I guess they're frowned upon for a reason, right? Like if people start describing everything based on one specimen and aren't able to look at that diversity within a species and get the idea around what species level divergences are, then it can make the process more complicated down the line. But I think if we don't start doing that, at least in some respect, then there's no way we can kind of go about this mission of describing everything.
Yeah, the question is whether we can adequately deal with that in a way that doesn't cause too much grief down the line.
Exactly.
So preempt some of the likely problems while not limiting ourselves necessarily. So the last question I had was a sort of a social one. So you're an ECR. So if you imagine a career stretching out ahead of you, where in addition to doing macroevolutionary studies and ecology and biology studies or whatever are the other things that arise from this, if in your career you're actually, in addition, naming a whole heap of new species because we've accelerated the pipeline, would you find that fulfilling, do you think, or do you think you would start quickly to find it very dull?
I mean, for me personally, that's what I enjoyed the most out of my PhD, and so that's what I'm trying to do the most of now in my postdoc. But it's very early days, like to go, could I do this every day forever? I'm not sure, but at the moment it's what I love most about taxonomy is the species descriptions. I think it's the piece of information that needs to come first, and it's the most useful and the most important to people. So for me, it's the most fulfilling bit is doing those species descriptions.
I think that you mentioned to me when we were chatting one time that you thought that most people who get into this game through university, one of the entry points and part of the joy of it is actually dealing with specimens and species and morphology and all of those things, descriptions.
Yeah, I think I mentioned that when we were talking about the possibilities around molecular species delimitation and that being kind of the be all and end all of taxonomy, and that I feel like so many people do taxonomy because they love the organisms. So removing that would be, I think, it would attract a different kind of person to the job.
But they also, of course, love having all of the molecular evidence that can then
exactly
doing the species limitation. So, yeah. Oh, well, look, Thanks very much.
No worries.
There was a beaut talk. I really liked it.
That's all right. It was a hard thing to put together coming from very limited experience. So hopefully it's useful.
I was really keen to get talks from people exactly like you, rather than just from the silverbacks in the game, because I think it's really important. So yeah, good on you, and thanks again.
Thanks, Kevin.
Good on you, Erinn.